For allogeneic CAR T cells, we've had several trials reported out. For example, Sean Melancote from our center was the first author on a trial based off the technologies developed by Selecta Celgene and Allogene, the product by Allogene, which did report efficacy. It has unfortunately been associated with toxicity due to the immune suppression strategy that they use to prevent rejection of the cells. Furthermore, while there is activity, I think the challenge of those trials has been to try to figure out how much activity is enough. We're all looking for a home run. We're all looking for a clear answer that allogeneic CAR T cells are better than autologous CAR T cells, and we definitely haven't seen that. So there is still an open question of how we design trials that will tell us when an allogeneic product is sufficiently active or more active than an autologous counterpart, such that it's worth developing further and eventually can become a standard therapy. In the in vivo space, we're very much in early days. We're just starting to see the first clinical reports of patients treated, mostly in China, but also in the United States. Reports of 3 to 4 patients with encouraging activity. We're already starting to see the first myeloma patients that have achieved stringent CRs with virally based approaches, referring to the recent report from SO Biotech. However, it is very early days. So we are really excited over the next year or two to see further reports in myeloma, in lymphoma, and increasingly in autoimmunity of in vivo CAR T cells.